Hello friends, welcome to my channel. Today's topic is first is eukaryotic cells, then cell membrane, then cell wall. Okay, first talk about eukaryotic cells. Eukaryotic cells contains membrane-bound organelles, and their compartmentalization increasing of and have complex cytoskeleton element and complex locomotor structure. And this is the structure and the shape of the eukaryotic cell. Then the network of protein of the filaments is three types. First is microtubule, which is diameter increasing diameter. Second is intermediate filament. Third is microfilament or actin filament, which is in decrease in diameter. Microtubule makes flagella motility. Now talk about cell membrane. It is also called plasma membrane or plasmolemma. This is a selectively permeable. This is protein plus lipid composition in a variable to from cell to cell. In mature mammalian RBC is used to membrane related research as it has no membrane bound organelle or no contamination in main membrane. Cell to cell, first is myelin sheath, lipid is 70 to 80%. Second is RBC, protein 52%, lipid 40%, carbohydrate and cholesterol 80%. Because due to less of contamination of RBC in the use to the sturdy membranes. Proteins on the basis of ease of extraction, this is of two types: intrinsic or integral, extrinsic or peripheral. Intrinsic is partially or totally buried, or embedded, or difficult to extract or difficult to or hard to remove. Extrinsic, easy to remove, or is the superficial. Now lipid. This lipid is a majorly phospholipid. This is a polar head hydrophilic, and this is non-polar tail hydrophobic, saturated hydrocarbon. Okay. Now the another point is in membrane, carbohydrate and cholesterol in animal is also found by side lipid and protein. In plants, other sterol like stigmasterol or composterol can be found. Okay. Now in 1972, Singer and Nicholson gave fluid mosaic model of plasma membrane, according to which membrane is a sea of lipids having iceberg of proteins. Okay. This is a extrinsic protein. This is a glycoprotein. Only on outside. This is a polar head. This is a neutral. This is a polar. This is the intrinsic protein. This is a lateral membrane. Cholesterol. This is a cholesterol membrane stabilizer. This is a glycolipid. First polar end. This water is present inside and outside the cell. Okay. And polar ends of both lipid layers face outwards, and this lateral movement within a lipid layer shown in by lipid and protein, and the flip-flop movement from one lipid layer to one lipid layer to another lipid layer shown by lipid in super exceptional cases never shown by proteins. This is a integral or transmembrane protein. Here non-polar or neutral solute high concentration to lower concentration through the simple diffusion and the water through osmosis process, and the polar solute needs carrier the facilitated diffusion from higher concentration to lower concentration, but against the concentration gradient increase to decrease to increase concentration active transport need ATP called pump active. Okay. The function is cell division, cell growth, transport, cell junction, secretion, exosis. Passive transport like diffusion, osmosis do not need energy, but active transport via pumps need ATP. Now talk about cell wall. Rigid, non-living layer present on the outer side of cell membrane. Rigid मतलब provide shape, non-living मतलब provide protection from the mechanical damage and the infection. It is the outermost layer. It helps in cell to cell interaction. It is a quite permeable but prevents the entry of very large macromolecules and let pass very small monomers. Some organisms and cell wall of the nature. Archaebacteria, pseudomurine, eubacteria, murin বা peptidoglycan, fungi, chitin, algae বা simple plant, cellulose, mannose, minerals like calcium carbonate, other plants, cellulose, hemicellulose, pectin, proteins. The function is shape and structure and the intercellular interaction. Now we can see in diagrammatically cytoplasmic standard. Okay, this is the young cell. This is the primary cell wall or flexible. This is the cell membrane. In maturation, the 1 degree cell wall is diminishing and the 2 degree cell wall inside the rigid or formed on inner side of 1 degree rigid cell cannot grow further. Okay, this is the cell membrane, and further the middle layer connects to the neighboring plant cell by the calcium pectate. This is called plasmodesmatas or cytoplasmic strand. Okay, this is the structure, or you can read. Now talk about endomembrane system. Endomembrane system. Inside eukaryotic cell, some membranous organelles become functionally coordinate and makes endomembrane system. This includes endoplasmic reticulum, ER, Golgi complex, lysosome and vacuoles. Okay, endoplasmic reticulum electron microscopy showed the presence of tiny tubular structure scattered in cytoplasm called ER. This is a two types, rough ER, RER or smooth RER and ACR. Rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough RER, ribosome present and will synthesize and secrete proteins, and ACR, absent of ribosome, will synthesize and secrete steroid and lipid. Now talk about Golgi complex. Camille Golgi discovered Golgi complex in 1898. There are many concentric flat parallel cisternae that makes Golgi complex. This type of structure. This is a flat structure, flat cisternae for forming cis convex. This is a median median Golgi, and this is a maturing trans or concave phase. Okay, the flow of the ER to cis to median to trans to target. Okay, this is the nucleus. Okay, this is a RER, this is a protein synthesis and secretion, and this is a ACR, this is a steroid and lipid, makes steroid and lipid, and this is a indirect contact of outer nuclear membrane, and this is a lumen of ER called luminal space, or everything other than the ER lumen is extra luminal space, and RER protein makes to go through the forming cis convex, then to mature transconvex this way, and then transphase to go this, and here depends about the sugar tag added during the glycosylation. The lipids and the protein are sent to intercellular and extracellular target, glycolipid and glycolipid formation of the packaging. Okay, then glycosylation occur in Golgi complex and packaging too. Okay, vesicle form Golgi complex makes lysosome. Here the hydrolytic enzyme work, and here pH is decreased because H plus concentration higher in this site, that's why pH is decreased. And then lysosome contains hydrolytic enzyme which is acid active at low pH 4 to 5. This is a single membrane. And then amylase which is a digest carbohydrate, lipase which is a digest lipids, okay, protease digest protein, nuclease digest nucleic acid, increase in acidity. And here the vacuole, the storehouse, H2O, SAB, extrude, etc. And no, here active transport because low concentration to higher concentration, and this is called tonoplast. In contractile vacuole in amoeba, food vacuole in protist. Amoeba has contractile vacuole that helps in the excretion and osmoregulation. Engulfed food in protist in covered by the thin membrane and food vacuole is formed. Okay. Today's lecture ends here. Thank you.